Hello all, welcome back to Dr. Kala Edu. Today we are going to discuss a topic on chromosomal mutations only. This is the continuation of the part 3. So chromosome mutation is divided into chromosomes in number and then chromosome in structures. So according to the type of mutation is divided. So numbers we have studied in detail in last two videos. We have discussed totally and regarding this euploidies and aneuploidies etc. So now regarding the chromosomal structures. So okay, generally when a structural variation in the chromosomes, we can tell same dialogue or same definitions. We can tell that something which is going to be added or deleted. Okay, so that means where in the chromosome structures, that is in the locations of the chromosomes itself, a mutation is created, then we call it as an chromosomal structural mutation, which is called as an structural chromosomal aberration, which is called as an structural chromosomal aberrations. Right? Okay, so what makes this chromosomal structural aberrations? So we can use lots of chemical compounds, ionizing compounds and radiations, both radiations and chemical compounds they cause these mutations. Okay, so here according to this, what is the thing is we can rejoin it. What is the thing? We can rejoin, or we can, exactly the word is reunion. Okay, so reunion of chromosomes occurs here in this structural chromosomal aberrations, right? So here according to that, we have mainly they have classified into two forms. We can tell it as an A and B. So the changes which is going to occur in the lossy, okay, the number gene of which is going to be there in the lossy. Lossy means, what is lossy? So a place where a particular gene is present is called as a lossy. Okay, right? So this is singular, and plural we can tell it as locus. Okay, so here generally is arrangement of gene lossy. So what here is number. So here there is going to be a disturbance in that particular chromosomal structures number, and here the arrangement of the particular gene is going to be getting disturbed, but it is not as like as gene mutations. So in gene mutations only a point, what we call it as an one codon, there may be a change. Hence we call it as a gene mutation. One particular gene is getting disturbed, that point. But here when it comes to chromosome, a lot of genes will be present in that particular chromosome and it is going to get disturbed here. So according to that number of the gene lossy, again it is divided into two forms. Okay, please be clear with this chart, don't get any confusions. So first, according to the number, we have a deletion, or which is called as deficiency. Obviously when a deletion takes place, what will happen? It causes deficiency. Hence we call it as also a deficient mutation, deletion or deficiency. Here we call it as an duplication or repeat. Why? A same gene numbers or same nucleotides, when it is repeated again, we call it as duplication. As when I am telling about the duplication itself, I use the word repeat. Okay, so hence we call it as repeat also. It may be called as duplication or repeat. So the word itself indicate that something is going to get eliminated. So here we are having two types of deletion. One is terminal deletion and another is intercalary deletion. So what is this? A simple way to explain you is, so we all know that all the nucleotides will be arranged as A, T, G, C, A, T, 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 C, C, G, G, like that and all. So for a general example, I am going to write you alphabets here. So for example, we have A, B, C, D, E and F. If it is arranged like this, see, when it is an terminal mutation. The word itself understood that. What is meant by terminal? It is an end. Meaning, I am giving the meaning as end. So what will happen? A may get deleted or F may get deleted. So this is called as an terminal end, that is terminal mutations. So what is intercalary? So intercalary is nothing but the middle, a C, or it may be a D. Okay, if the deletion happens in the middle, then we call it as an intercalary mutations. I think here it is simple and clear. So example here for these type of mutations are seen in Drosophila and maize. Commonly it is seen in Drosophila and maize, right. So now is this duplication or repeat. So this duplication, actually it was first reported by Bridges. The scientist name is Bridges in the year 1919. Okay, in the year 1919 only he reported duplications, right. So where from which organism? Drosophila. So lots of experiments were done only using Drosophila fruit flies. Much more experiments regarding linkage and then recombinations, mutations, all the experiments were done using Drosophila. Okay, so duplications, here we see come across three types are tandem, reverse tandem and displaced tandem. Okay, so it is displaced tandem. So three types here: tandem, reverse tandem and then displaced tandem. So here also I will explain as like this in a simplified manner. So here in tandem what happens is A, B, C, D, E, F, etc. So let us consider the same letters as like here. So the B and C is getting duplicated. So duplicated means what? It is going to have another copy of it. So what will happen here? In tandem what will happen is, so A, B, C, again B, B, C, D, F. So it is getting duplicated here. So this is called as tandem duplication. And next is reverse tandem. So reverse tandem what will happen? Same we can continue. So A, B, C, but it is going to get revert, that is C, B, E, F, like this. Immediately it will get an reverse position which is called as an reverse tandem. And then displaced tandem. In displaced tandem what will happen? It is going to displace somewhere in the different loci, that is it may be A, B, C here. This is going to get duplicated. But it is not going to be placed immediately after that. The duplications is going to be placed something in different place. For example, all after this again B, C, and it is duplicated, displaced here, right? This duplication is displaced later after some of the genes location. So here of three types. So now changes in number gene loci is completed. Next is arrangement of gene loci. So in arrangement we have again two types. One is inversion and then is translocation. So the word inversion means we are going to get something is rearranged. So here what happens is the chromosome itself will get rearranged. That is here rearrangement takes place in inversion. Okay, rearrangement of chromosomes. Okay, so when will this happen? When the DNA is getting break somewhere, when we are inducing mutations, when the DNA is going to getting break, this type of inversions takes place. There are two types of inversion. One is paracentric and another is pericentric. So here the paracentric, only one arm of the chromosome is going to get inverted. Only one arm of the chromosome is going to get inverted. So hence it is paracentric. So the rearrangement will be in that only particular arm. But when it is pericentric, there will be an involvement of centromere. So there will be an there will be an involvement of centromere because any two arms of the chromosome is going to be in inversion, so that there is an help of that centromere while there is a rearrangement of the genes, right? So next is translocation. So translocation, what will happen here is the location of that particular gene or that particular chromosome, the chromosomes will be as translocated, the place will be changed. Okay, so trans and then location, the location is changed. But here the one thing you should not get confused is crossing over is exactly different from this translocation. So in crossing over, it is a natural event where during the meiosis the process is taking place. But in this translocation has happened due to the induction of mutations. So what will happen? A small break in that particular chromosome, and then it is going to join with another chromosome, which is called as a translocation. So this may be, we can shortly tell as interchange of chromosome segments. So what is that? Interchange of chromosome segments, which is called as an translocations. So here it is of three types. One is simple, reciprocal and shift translocation. Okay, simple translocations, what will happen is a change in that particular location, nearby location itself, the chromosomes and it's getting changed, which is called as a simple translocation. And then reciprocal translocation, the word itself indicating that one piece of chromosome will be cut and it is passed to the near one, and from there it is going to get reciprocal here, which is called as a reciprocal translocation. That is, exchange of the chromosomal parts is called as a reciprocal translocation. And then shift translocation. So in this shift translocation, what will happen is segments itself, the segment of the chromosome itself will cut and it is going to shifted to the near or shifted to be in the same chromosome, or it may be nearby another chromosome also, which is called as a shift translocations. So this is all about that, how the chromosomal structural mutation has been happened. So we have, according to that exam point of view, you should know all this terminal, intercalary, and then duplication and repetition, translocations. So everything want to be clear, and especially you can get here 2 mark questions like what is tandem, what is reverse tandem, what is displaced tandem, like that. So you are clear in this chromosomal mutations, I think. If you have any doubts, you can comment me in the comment section. Thank you. Meet you in the next video.